So good morning everyone and welcome back to our channel. So today we are going to talk about genes, continuing our previous chapter. Genes जो हैं वो आपके DNA पर present होते हैं and they are the specific sequences of nucleotides. इनका मेन फंक्शन है DNA को मैसेज पहुंचाना या मैसेज देना कि वो प्रोटीन का सिंथेसिस करे. और सबसे इम्पॉर्टेंट जो इसका रोल है दैट इज़ इन द ट्रांसफ़र ऑफ कैरेक्टर्स फ्रॉम पेरेंट्स टू दी ऑफस्प्रिंग्स which we will study in the next chapter in much detail. So let's see that why do we need new cells, what is the need for new cells. Basically 4 मेन रीजंस हैं जिसके लिए हमें न्यू सेल्स की ज़रूरत होती है. पहला, ग्रोथ. आपकी बॉडी में जो ग्रोथ हो रही है वो नए सेल्स की फॉर्मेशन से हो रहा है. Second is repair. When any of your part is damaged, any tissue is damaged, so from cell division new cells are formed and those parts are getting repaired. Third is a replacement. For example, as given in your book, in every second, millions of RBCs are getting destroyed or getting replaced by new RBCs. This is called replacement and that takes place through cell division, that is formation of new cells. And last is reproduction, which is related to meiosis in you can say higher organisms, and in some of them like asexual organisms we have fission, nuclear fission, like in amoeba you have studied. So in meiosis in higher organisms like human beings, what happens that the gametes, or you can say the sperms and the ova, are formed and later on by their fusion the zygote is formed. So they are also formed from cell division. Now let's come to a very important topic called cell cycle. Cell cycle क्या है? Cell cycle is a series of events. बहुत सारे events इसमें होते हैं और इसका main जो purpose है that is कि new cells are formed and the DNA is getting duplicated. Along with the duplication of chromosomes, and 2 daughter cells are formed here. So here cell cycle has 2 main you can say phases or parts or steps. The first is interphase, second is M phase or mitotic phase. Let's see what are they. So interphase जो है, this is the resting phase of a cell. What happens that when cells are formed, for example when 2 cells are formed, so they are quite small from their mother cell or their parent cell, and the nucleus is full sized, but the cytoplasm is very less because of the full size of nucleus, and also the cell is very small. So during this time the cell becomes larger and increases its cytoplasm and becomes ready for cell division for formation of new cells. So why this phase is called resting phase? Because there is no change in the chromosome number. But although it is called a resting phase, there is quite active synthesis of DNA during this phase. This phase again is divided into 3 main phases, and the first is your G1 or first growth phase. In this phase, RNA and protein is getting synthesized. Along with that, the volume of cytoplasm increases, and in animal cells mitochondria and in plant cells chloroplast, these two divide and duplicate themselves. They are also called as semi-autonomous cell organelles because they have their own DNA and therefore they can replicate. And here the cell decides whether it wants to get divided to form new cells or it again wants to go into the resting phase. It decides this. So if it wants to divide, it will go to the S phase or the synthesis phase. Coming to S phase, so basically 2 main things happen here. First is that DNA is getting synthesized, and second is that the chromosome is duplicated but they remain attached to each other. Now last is your second growth phase. So second growth phase, what happens that RNA and protein synthesis is continued, and here the cell is ready for the cell division. Now after the cell division, when the new cells are formed, they will again go to the interphase, and again they will become larger and then G1 phase, S phase, G2 phase. In this way, this cycle continues. So this is what you need to focus. Now, formation of the new DNA. Just now we studied that in S phase new DNA are formed. How does this happen? So here what happens, that DNA, it replicates. So how it replicates, as you can see in the picture, that from one side the DNA strands open, helical strands, they open, and to these strands, to the old strands, new strands start to form, and then they divide, and as you can see, two DNAs are formed. So in this way the DNA gets duplicated during the S phase of the interphase. Now let's see the next. Now question arises that does this cell division continues forever? So the answer is no. At some places it stops permanently, at some places it is temporary, and at others till it is needed. In children, like we children, cells are continuously they are produced and they replace the old cells. So what happens, that the number of and in what happens in adults, cell population stays constant in the sense that the number of cells which are dying, they equal the number of cells which are produced. But in old people, the number of cells produced are less than the number of cells dying. Okay, so this you have to keep in mind. Now, cell division. So we know that there are two types of cell division, mitosis and meiosis. What is the main point here, that in mitosis, same number of chromosomes is passed to the daughter cells. But in meiosis, haploid number means half of the number of chromosomes is passed to the sperms and ova. And in mitosis, diploid number of chromosomes is passed. Now just you can see here, I will not, this is the last slide. You can have a review that what we are going to study in the next video. Mitosis has four main phases. Actually, not four. First, it has two phases: karyokinesis and cytokinesis. So in karyokinesis, these four phases are there: prophase, metaphase, anaphase, and telophase. You can see in the diagram little bit about these phases. So we will discuss these phases in the next video. So till then, stay healthy, stay safe, and we will meet in the next video. Thank you very much.